Hello and welcome back to part 2 of chapter 6. Last lecture we focused on metabolic pathways with an in-depth look at cellular respiration. In the second half of chapter 6, we will focus on photosynthesis. As I emphasized last lecture, I will emphasize to read the chapter and take your time with the material. Photosynthesis, just like cellular respiration, has some complexity to it as well. With that out of the way, let's jump right in. As we will learn about photosynthesis, we will see that it is linked to cellular respiration. The byproducts of photosynthesis, glucose and oxygen, go into cellular respiration, while the waste products of cellular respiration, carbon dioxide, is used within photosynthesis. This linkage occurs frequently in plants as they both have cellular respiration and photosynthesis. Organic molecules produced in these reactions can enter catabolic or anabolic pathways. These relationships comprise and make up a metabolic system. Figure 6.13 illustrates this point that metabolic pathways are linked. As we learned about cellular respiration in the last lecture, products of one reaction are often reactants for the next reaction. Last lecture we focused on the oxidation and reduction of glucose to make ATP. But lipids can break down to form acetyl coenzyme A, which can directly go into the citric acid cycle, while proteins can be broken down to amino acids that can be used in glycolysis, or the citric acid cycle. ATP and reduced coenzymes link the catabolism and anabolism in cellular respiration with photosynthesis, as we will see in a little bit. Photosynthesis and cellular respiration are linked by their reactants and products. On this slide we have two equations that summarize cellular respiration and photosynthesis. In cellular respiration, glucose is oxidized to make ATP, chemical energy, and produces carbon dioxide and water as byproducts. Photosynthesis converts light energy to make chemical energy. Photosynthesis takes carbon dioxide, water, and light energy to produce carbohydrates, such as glucose, and oxygen is produced as a byproduct. Figure 6.14 is illustrating how photosynthesis and cellular respiration are linked, with their products going from one to the other. The coenzyme ATP is the final product of cellular respiration. As we will see, ATP is needed to drive photosynthesis. Photosynthesis is an anabolic reaction, meaning it is bringing molecules together to store energy. Photosynthesis can be broken into two pathways: light reactions and carbon fixation reactions. Light reactions is where light energy is converted to make ATP and produces the reduced electron carrier molecule NADPH. NADPH is similar to NADH, which you find in cellular respiration. But please note that NADPH and NADH are two different molecules found in two different metabolic pathways. The carbon fixation reactions, which occur after the light reactions, use the ATP and NADPH that are produced from the light reactions to eventually make carbohydrates. As we will see, photosynthesis takes place in the organelle chloroplast. Light reactions occur in the thylakoid membranes, while the carbon fixation reactions take place in the stroma of the chloroplast. Before we get into the details about photosynthesis, what makes light a suitable source of energy for plants? Light is a form of electromagnetic radiation. It moves as a wave, but behaves as a particle, like photons. Photons can be absorbed by specific receptor molecules in chloroplasts and raised to an excited state that has higher energy. Pigments are molecules that absorb wavelengths in the visible spectrum. Chlorophyll absorbs blue and red light. The remaining light that is reflected back at us is mostly green. You can see in figure 6.17 that this plant absorbs best within the red and blue spectrum of light, using the pigment chlorophyll. Plants have two chlorophyll pigments that absorb light energy, chlorophyll A and chlorophyll B. There are some accessory pigments such as carotenoids and xanthophyll. These accessory pigments absorb wavelengths between red and blue and pass that energy to the chlorophylls. These accessory pigments are colors you see in the fall as the green chlorophylls begin to break down. The pigments are arranged into light harvesting complexes that act as antennae for light energy. A photosystem spans the thylakoid membrane in the chloroplast. The photosystem contains multiple antennae systems surrounding a reaction center. Figure 6.19, shown here, illustrates what a photosystem looks like. In the center, you have the reaction center surrounded by light harvesting pigments. Those pigments will absorb and transfer that energy to the reaction center. When chlorophyll absorbs light and enters an excited state, becoming excited chlorophyll. When the excited chlorophyll returns to a ground state, it releases an excited electron. The excited chlorophyll gives the excited electron to an acceptor and becomes oxidized, since it lost that electron, becoming chlorophyll plus. The acceptor molecule becomes reduced. At this moment, the reaction center has converted light energy to chemical energy. Here in figure 6.5, we see that a photon from light energy is being captured by the chlorophyll molecule, causing an electron to become excited from the reaction center. And that electron is then transferred to an electron acceptor. The electron acceptor that received the electron from the photosystem is the first carrier. An electron transport system occurs in the thylakoid membrane. The final electron acceptor is a NAD+, which gets reduced to an NADH. In addition, ATP can be produced during electron transport. Figure 6.20 illustrates what is happening in these photosystems. Here we have photosystem 2 and photosystem 1, with electron transport occurring between the two photosystems. Electron transport is used to produce ATP and then NADH. These are the final electron acceptors in the light reaction. We'll go over these photosystems in more detail. Within the light reactions, there are two photosystems, photosystem 2 and photosystem 1. Photosystem 2 occurs before photosystem 1 because photosystem 2 was discovered after photosystem 1. And rather than renaming or renumbering them, they wanted to make our lives more difficult. So please bear with me here. We'll start with photosystem 2. Photosystem 2 absorbs light energy, oxidizes water, and initiates ATP production. Photosystem 1 absorbs light energy and passes an excited electron to NAD+, reducing it to NADPH. Both photosystems occur in the thylakoid membrane of chloroplasts. We will look at both photosystems in a little more detail. In photosystem 2, when the excited chlorophyll gives up an electron, it becomes unstable and grabs an electron from water to replace it, thus splitting water into oxygen. This is the reason we can breathe, because photosystem 2 needs an electron from water. Because of this splitting of water in photosystem 2 is the reason why we get oxygen. So please be sure to thank a plant today. The excited electron from photosystem 2 is passed through a series of thylakoid membrane-bound carriers to a final acceptor at a lower energy level. While this is happening, a proton gradient is generated and used by ATP synthase to make ATP. Very similar to the electron transport chain in cellular respiration. The electron that is being passed down this electron transport chain is eventually passed to photosystem 1. Within photosystem 1, there are two scenarios that can occur with the electron that gets excited. There is non-cyclic electron transport and cyclic electron transport. We'll talk about the non-cyclic electron transport first. Photosystem 1 also receives light energy, just like photosystem 2. When the chlorophyll in photosystem 1 becomes excited, the electron will leave and end up reducing NADP+ to NADPH. The electron that leaves the chlorophyll must be replaced, and it comes from the last electron carrier in photosystem 2, thus the name non-cyclic electron transport. In cyclic electron transport, again, occurs within photosystem 1, ATP is produced, and no NADPH is produced, like in non-cyclic transport. This ATP is needed in the carbon fixation pathways that come next. Then cyclic transport, an electron is passed from excited chlorophyll through the electron transport chain. However, instead of this electron going to NADPH and leaving the cycle, it recycles back to the same chlorophyll to be used again. This electron transport chain produces ATP. Figure 6.21 illustrates the cyclic transport photosystem 1. As the chlorophyll becomes excited, it ejects an electron. That electron is then passed down electron transport chain, producing ATP. That electron then eventually returns back to that chlorophyll. Again, this is cyclic transport, meaning the electron never leaves the photosystem. This only occurs within photosystem 1. Okay, we went over a lot of exciting material. Please review anything that was unclear to you and do your best to answer these progress check questions before moving on with the lecture. Okay, so we just learned about the light reactions in photosystem 2 and photosystem 1. Next major metabolic pathway in photosynthesis is carbon fixation. And again, carbon fixation occurs after photosystems 2 and 1. Calvin cycle is the metabolic pathway that will use ATP and NADPH that are produced from photosystem 2 and 1 to fix carbon dioxide and make it a reduced molecule in the form of carbohydrates. The Calvin cycle occurs in the stroma of the chloroplast. Each reaction in the cycle requires a specific enzyme, and the cycle can be split into 3 distinct processes. The 3 stages of the Calvin cycle are fixation of carbon dioxide, reduction of 3-PG, and production of G3P in regeneration of RUBP. Here's a general diagram of the Calvin cycle. We'll break it down mostly to the products of each step. However, I want you to make note that you can see that ATP and NADPH that participate in these reactions. Their role is to help fuel the cycle with energy from the ATP or in the redox reactions from NADPH. This is what links the light reactions with the carbon fixation reactions. Without the energy produced from light reactions, the Calvin cycle wouldn't occur. First step of the Calvin cycle is the fixation of carbon dioxide. CO2 is added to the molecule ribulose 1,5-bisphosphate, or RUBP for short. The enzyme that catalyzes this reaction is ribulose biphosphate carboxylase oxygenase, or you can just call it Ribisco for short. During this reaction, there's a formation of a 6-carbon molecule that will eventually break into 2 3-carbon molecules called 3-phosphoglycerate, or 3-PG for short. Figure 6.23 briefly goes over the fixation of carbon. CO2 binds with RUBP, with the final product being 3-PG. The second step of the Calvin cycle uses the 3-PG from the first step. 3-PG is reduced to form the molecule G3P. This step is appropriately titled the reduction of 3-PG. The final step of the Calvin cycle is the production of GP3 and regeneration of RUBP. The molecule G3P produced in the last step is used to make RUBP so that the cycle may continue. Some extra G3P that is produced will leave the Calvin cycle. This is the important part and why we're going through all this. The G3P that leaves the Calvin cycle is important because it can be used to produce glucose and make starches and cellulose, or things that can be used in cellular respiration to break down to make ATP. The energy provided from carbohydrates produced by the Calvin cycle provide almost all the energy for life on Earth. Photosynthetic organisms, also referred to as autotrophs, use most of this energy to support their own growth and reproduction, meaning plants aren't necessarily producing this for us. They're using it for their own needs. Heterotrophs, such as ourselves, cannot photosynthesize, and we depend on autotrophs for chemical energy. That's why we're learning about photosynthesis. Okay, last progress check to end the chapter. Please take your time to learn the Kelvin cycle. Focus on what products are produced where. Do your best to keep your RUBPs, 3PGs, and G3Ps straight from one another. Unfortunately, many students find this material rather dry. Not everyone likes photosynthesis, which can be hard to relate to. So with that said, I want to end on a joke. If a plant is sad, do the other plants photosynthesize with it? I think we can all relate to that. Anyways, thank you for listening. That wraps up Chapter 6, and have a good rest of your day.